Hi, Dr. Wang here. Now we're going to do assignment APE5. This assignment used the same set of sequences as APE4, and I have these two files already opened on APE. So now let's look at the actual question. In the first question, this assignment asks us to identify a pair of primers that you can use in PCR to amplify a fragment that contain the mutation in this mutant plasmid. So before we can figure out which primer pair is going to work, we need to know where this mutation is. So we have the wild-type DNA sequence here and the mutant DNA sequence here. We can simply align the two DNA sequences. This is going to tell us where the mutation is. You can, of course, do this in cluster omega, but APE has a built-in alignment tool that will allow you to align DNA sequences. So go to Tools, Align Two Sequences. And then over here, I'm going to pick the wild type PMSH2 and then align that with the mutant version. All right, so click OK. So here, this is the alignment outcome. So as you can see here, the two sequences are mostly identical. So if you scroll down, you will see that the mutation is highlighted in red. And it is right here in location 3,800. So it's a T2G mutation. So now I'm going to find this location on my mutant plasmid. So I can do so by copy the sequence just in front of this mutation. Come back to the plasmid sequence, go to edit, and then find, and then search for this sequence. And we can see that it's right here. This G is right here, and its location is 3,800. So now I'm going to go to Features, New Feature, and then call this mutation. So now we have this mutation highlighted on the mutant plasmid. So the next thing we're going to do is to find the location of the primers. So I'm going to make this a little bit larger. Okay. Okay, so now we are going to find the 4 primers. It's pretty straightforward. Just copy the sequence, come back, and then do a find. So here we can see that APE have discovered this sequence. So we're looking for ATTT, and it discovered ATTT. So this sequence means that this sequence is located on the top strand. In another word, this is a forward primer. So I'm going to call this primer 1. And because this is a forward primer, we're going to leave this box unchecked. So I'm going to say, okay. All right, so what I'm going to do on the side is to actually draw this on a piece of paper. So we have the entire sequence, right? So the mutation is right here. And primer 1, let's see where is the relative location for primer 1. So mutation is right here in location 3,800. And then primer 1 is a forward primer, and its location is right here. So it starts at 4,000. So its relative location is right here. So I'm going to draw this primer. So this is what primer 1 looks like. Now we're going to look for primer 2. So it's the same step, copy, and then find. So here we're looking for TTCTT, and we found TTCTT. So this is a forward primer as well. And we can see that it's located before the mutation. All right, so I'm here going to call this primer 2, and then also leave this box unchecked because it's also a forward primer, and its relative location is right here. So this is where primer 2 is. And I'm going to repeat the step for primer 3, copy the sequence, come back, search, and find next. So here we are looking for GATG, and then we found ATAT. So the two sequences are not identical, and actually they're reverse complementary to each other. So this is a reverse primer. All right, so to make this a feature, I'm going to call this primer 3. And then check this box to indicate that this is a reverse primer. And we can see that it's located just right before where this mutation is. So I'm going to come back and then draw its relative location. So primer 3 is actually right here, just slightly before where the mutation is, and it is a reverse primer going the opposite direction as primer 1 and 2. Now we're going to find primer 4. Search, find next. All right, so here we can see that this sequence and this sequence are reverse complementary to each other. They're not identical. So here primer 4 is a reverse primer as well. And it's located somewhere in between this primer and the mutation. All right, so let's remember that. So I'm going to close and then make this a new feature called primer 4, and this is a reverse primer. Okay, so and its relative location would be between the mutation and primer 1, and it's going in the reverse direction. So this is what primer 4 is. All right, so here we have everything labeled and also illustrated right here. So now the question asks for, so which pair of the primer is going to work in PCR? Right, so for a primer pair to work in PCR, they have to be running towards each other. Right? So this primer pair is going to work. And then this primer pair, they're running away from each other, so it's not going to work. And similarly, this is not going to work, and this is not going to work either. So none of those will work. And then also we want to amplify the mutation, right? So we want to make sure that when this pair of primer worked in PCR, the mutation is located right in the middle. All right. So the which pair of primer will allow you to do so? There's only one pair. All right, so now we're going to do the second question. So the second question asks you to use this pair of primers that you have identified in the first question to perform PCR. So I'm actually going to give you the right answers here. So the pair of primer that works is right here. Primer 2 and primer 4. We can see that primer 2 and 4 run towards each other. Right? So during PCR is going to work this way. And then here's our PCR product. So pay attention that the PCR product actually includes the sequence of the primer. So how long this PCR product is, all you have to do is to go from the beginning of primer 2, which I believe is right here, right? So primer 2 is right here. So highlight the entire sequence all the way from the beginning of primer 2, go all the way down to where primer 4 ends, and then I'm going to copy and then here open a new window. I think I opened the wrong thing. Okay, sorry. Go to File, New, and then paste that sequence right here. So we can see that this is the actual PCR product, right? So how many base pairs do we have in here? We can see that this mutation is actually right in here. So we can now save this file. So I'm going to save this and then give it a different name. I'm going to call this PCR mutant because we actually simulated the PCR reaction using primer 2 and 4 and using the PMSH2 mutant as the template. So I'm going to call this the PCR mutant. And I'm going to do the identical steps for PMSH2, right? So just do the same thing for PMSH2. Not the mutant, do this for the wild type, and then you should have another file called the PCR wild type. So those two files, we are going to use them in the next APE assignment, APE 6.